Give me a second to start.
Tell me when.
Alberto, it's all yours.
Okay, thank you. Well, thanks, Piero, for the invitation. I actually have never given a talk for such a broad audience, so this is a first. Let's see. Hopefully, I hit the right notes. This talk is on my research area, which is the use of polymers that are inherently soft for bioelectronics, or one of the areas of my research. And the reason why one would be interested in that is that there is a lot of interest lately in interfacing electronics with biology. So if you think of the human body, you want to sense and diagnose what's going on. This is the whole idea behind precision medicine. But you could even go one step further, and then using that data to actuate therapy, which could be actually electricity itself could be a therapy, or electricity could be the vehicle to deliver a therapy. And so if you want to interface biology with microelectronics, there is some fundamental mismatch. Biology is typically soft, what is called a Young's modulus, so the hardness, not the hardness, but how the materials react to external stresses in biology makes materials soft. Microelectronics typically is made with stuff that is hard, like rocks, silicon. Biology is typically adaptive. It changes continuously according to its environment. Once you program a computer chip, you can certainly change programs, but the chip itself, its architecture is fixed. And then finally, biology works mostly by using ions and biomolecules, and all electronics works by using electronic charges, that are called electrons and holes. And so there is a whole family of materials that are both soft and have the properties, the electronic properties that you require, and these are called organic semiconductors. So they're made of molecules that are a little bit similar to the molecules that you find in the human body in terms of mechanical properties, but then have the electronic properties of semiconductors that you would use for electronics. So here's an example. This is actually, in order to be able to interact with biology, you have to be able to interact with the ions. So you have to have the electronic part, that's the electrons. And so here is a material that has one polymer strand that has the electrons in it, and you just blend it with another strand that likes ions. And it's sort of interesting, when you put these two together, they form structures a little bit like, in proteins you have the primary, secondary, and so on. They end up forming, when you cast them into a film, these structures where you have pancakes of electronically active regions, and in between you have these other regions that like ions. And so when you put this in water, water kind of permeates everything here, kind of like a sponge. So you have a sponge that contains ions, but at the same time can conduct electrons. And so you can imagine how this is a nice way to interact with the human body that will exchange ions, and those ions will interact with the electrons and modulate the electronic conductivity of this material. And so it's a way of transducing the body signal, which is ions, into the signal that electronics can understand, which is electrons. This is one way of doing it. Here's another way of doing it, is to design a polymer that has one part that conducts electrons, and on the same polymer, you graft another part because of these oxygen ions, these oxygen atoms. This will like ions that are in an electrolyte, so it's salty water, which is what we're mostly made of. And the reason why I'm showing this is because it showcases an important aspect of polymers, which is you can kind of design them a little bit the way you want. Oh, I want something that's electronically conducting. Here it is. And then I can graft on it something that's ionically conducting. So for us in material science, this is exciting because it's sort of a materials by design family where in principle, if you understand the rules of how you make the material that you want to have the properties, the desirable properties, you can just go out and make it, which is unique in nature. You get whatever nature gives you. Here you can synthetically make what you like based on the properties that you want, provided you understand how the structure is related to the properties. And that's the bulk of my research, and we still don't understand how the structure is related to the properties. So we sort of make materials by trial and error. Even then, we're pretty successful in doing that. So if you have now this polymer strand that has ions around it, it will just not stay straight like this. It will start coiling, and eventually when you form a film, you can see how now the ions and the electrons are intermingled close together. They're coupled, and so when you start exchanging ions with the human body, these will be coupled to electrons, and I can measure the electronic response. And that's how I have this transduction from the human body to an electronic device. And eventually this looks like this. When it's hydrated, you have here the highways of electronic transport, and then in between you have all the water with the ions. So you really can think of an interconnected sponge of electronic highways, and then there's ions and water in there, and that's how they communicate. So that's a general principle of how the material works. And so this is kind of a brand new way of interfacing with a living matter, because until now, if you wanted to interface electronically with living matter, you have this rock, and of course the surface, when you put it in water, oxidizes a little bit, maybe it softens a little bit, but now you actually have something that can be in intimate contact with whatever physiological fluid you have, because this will come in and interact with the semiconductor throughout all of it and couple to it very strongly. So it's really a different way of having semiconductors. We also call them porous semiconductors because they can accept physiological fluid in it as if it had pores, even though they technically don't really have pores. They're more, I like to think of it as a sponge. And so the fact that you have both ionic and electronic transport allows you to emulate biology. So if you think of a cellular membrane that through ion channels will traffic sodium or potassium ions and so on. So in this case, for example, the neuron, when it spikes, you have sodium ions that go in, and then it polarizes, and then potassium ions go in and out, and so on and so forth. So you can see essentially at a high level the way this neuron works. It works by exchanging ions with the environment. And I just told you our semiconductors do exactly that. They exchange ions with the environment, and then they'll produce an electronic signature of that exchange of ions. So you can imagine now one of these materials that, as I said, is an ion sponge, contains them here, and then if you put, for example, if you want to push the positive charges in this direction, I put a positive potential here, positive and positive repel each other, so this pushes the ions through into this other material. So you can see it's a little bit similar, right? You're pushing ions across a certain region. And you can do even better. One very fundamental property of the synapses, which is the space that connects neurons, is what is called potentiation. So the more signals exchanged from one neuron to the other, the more the strength of this connection increases, because essentially more of these receptors for the neurotransmitters appear on the surface of the postsynaptic neurons. In other words, the more one neuron signal to the other, the better that connection, and that's sort of the fundamental mechanism of memory. You reinforce memory by always connecting the same neurons one to the other, and that's called long-term potentiation. So you can emulate that with our materials as well, because I can keep pushing ions from here into there. So if I push a cation here into there, it has to be compensated by an electron for electron neutrality, and so I'm charging this material more and more. And I could call this charging potentiation. So you can see how the material will have a memory of how many ions I pushed. Eventually the ions leak out, and so this is long-term potentiation, but with a certain lifetime, which is exactly what happens in your memory. So you can see just in the material you can emulate an important aspect of the human brain, which is a synaptic connection between neurons. Now, why is this interesting? Well, we all know that the energy requirements for computing are essentially blowing up, because we all want to have really complicated AI models to recognize images of cats and dogs and so on and so forth, and other fundamental problems. And so we're looking at ways to do computation with low power. So here's a really old plot and a paper that shows that if you look at the power density of chips, it keeps going up. And a modern chip has more than 100 watts full power dissipated when it's going at its maximum power, which is like an incandescent light bulb, right? And that's what's sitting on your lap. On the other hand, the brain is slow, but does amazing things even though it's slow and uses very little power. And so people are interested in emulating and computing how the brain works. One of the fundamental reasons why chips use so much power is what is called a von Neumann architecture. Essentially, computing and memory are separated. So you have your transistors here, you have your memory there, and you're continuously shuttling data back and forth, and that uses a lot of power. Your brain, on the other hand, has computing and memory in the same unit. The synapse and the neurons do both computing and memory. And so co-locating computing and memory is one of the ways to reduce the power of computation. And you can do this actually very elegantly. If you think of a neural net, which is sort of what everyone uses nowadays to do to do artificial intelligence type calculations. The way a neural net works is a little bit like your brain, where you have a neuron. The neuron receives signals from all these other neurons that are connected to it through synapses. And remember, the synapses, the more they fire together, the more the signal gets potentiated, right? And so some signals potentiate, some signals depotentiate. In the end, you form a network that is able to produce the result that you want by training. And that's what you do in a neural net. So you have an input. Imagine this input, for example, is 128 pixels. And the output, you want it to be a digit from 0 to 10 to 9, right? So you have 128 inputs, and you have 10 outputs. And what you want is you want to recognize a digit in that pixelated image. And so what you do is you have all these inputs, and then you connect it to different layers, where each layer plays a role, each one of these dots plays a role of a neuron, where the signal from all the previous layers comes in here with a weight, and then you tweak the weights until your network recognizes the digits. That's called a training phase. Then you can use it. It will recognize, you have a training set. Once it's trained, it will recognize all the digits after that. So the key for this to work is really these weights that have to be tweaked, and then once they're tweaked, they have to stay where they are. And currently, you do this in software, and it uses a lot of energy. But you could do this in hardware just using physical laws. If I make what is called a crossbar array of wires, horizontal wires and vertical wires, and I connect them by a resistor. So at this node, what is called Ohm's law will say that if I have a voltage here, and this node has a certain conductance, voltage times conductance is current. And then if I sum all the currents, it's essentially what happens each one of these. It's the signal at each of the inputs multiplied by the weight. And the weight is this conductance. Or in other words, you can replace a layer of this neural net by a crossbar array in hardware. And this will do essentially the computation for you for free, just because of physical laws. So you send in your inputs, and your outputs will be the result exactly what you would have in the neural net. You change the weights, so you change the conductances, and that's how you train your network. So this will allow you to do what a neural net does in parallel with very low power. And doing these type of operations is one of the basis of a lot of artificial intelligence computations. And so this is what we're trying to do. We're trying to make these play the role of synapses, right? They're the thing that gets potentiated or depotentiated when you have a signal going from one neuron to the next. So we've been making what are called artificial synapses using our polymers. Essentially, it's this device. You send in ions through it, and then if you have more ions, the conductance changes, you're potentiating. If you send fewer ions, the conductance decreases, you're depotentiating. This is what the device looks like. You operate it, you send the potential, this sends ions across this region. They have to travel across, they go to the other side, they change the conductance of this channel, and that's it. You've modulated, essentially, the conductance of the channel. Your synapse has been activated. And then you disconnect it, and once you disconnect it, nothing happens, because there is no path for electrons to go back or for ions to go back. Essentially, this is like a battery that's sitting on your desk. So it's kind of interesting. Something that has a memory for information, it's storing information. It's the same thing as if it was storing charge, and that's what a battery does. So if you think about it, it makes sense. So we've been making these synapses. This is what they look like once you fabricate them. And then you can actually make them so that they operate very fast. The only thing I want you to notice here is the time scale. So we're changing the potentiating and depotentiating the synapse. You can see we can do it in microseconds, because just the same principle used in microelectronics, you shrink things down, they become faster, and they also use less energy. So if you look here at the switching energy per unit switch, we're actually down to 80 femtojoules. So that's a tiny amount of energy, very comparable to a biological synapse, which is, again, remember, our goal is to make things that use very little power. So this seems to be working in that direction. So we have something that can work in parallel, similar principle at your brain, and uses very little power. So we've used this to train what is called a model, logic gates. Essentially, to not go into detail, you have two inputs, and then you have your classical AND, OR, and NAND logic operations. So you have your inputs, and your outputs could be anything, but you can train your system to make it so that the first output is an AND, the second output is an OR, and the third output is an AND. And essentially, you can train this little array to learn how to do these three basic logical operations. But the problem is that this is what it looks like, right? It's incredibly complicated in terms of wiring, so we have a lot of work to do to make this actually something useful. But this is, even though it's only a 3x3 array, so it's small, it's 9 devices, you can already tell the complication of the wiring. But you can actually do something simpler and kind of cooler, which is what we call in-material learning. So you can, this is what we did by using a little Lego robot. Our device can remember. So what we did is we said, why don't we use it to train a robot to do a very simple task? That task will be to navigate a maze. Let's see if I can run the video. So here's the learning. Every time the device sees one of these and needs to make a left, sorry, the robot sees one of these and needs to make a left turn. And if it doesn't see it, it needs to make a right turn. So you see when it starts, okay, it's learned here, so it's going well. It's learned here, it's going well. Then when it hits this one, it takes a wrong turn. So what you do is every time your robot takes a wrong turn, you tweak your synapse a little bit, so there's a bit of learning, until it takes enough turns that are the right turn that it's learned, and now it can go navigate the maze. So the learning really is in the material, right? This little channel of polymer is remembering the lesson. It's being punished every time the robot takes the wrong turn. It's remembering the lesson, and eventually the learning is in the material itself, or in the charge that's been stored in the material. And eventually, successfully, this robot can navigate the maze and get out thanks to the cues that it's being given. And once it's learned, we'll be able to put these cues in any position, and it will always take the turns that it's supposed to take to navigate the maze. So it cannot navigate it by itself, but it's learned to read the signals that we gave it. And the learning really is in the material. Now, I said one of the other things that's nice is that these are soft, and so they're compatible with living matter, so you can put them on the surface of the brain without the brain being damaged. Here, actually, they've been inserted, and the reason why you don't see where they're being inserted is that there's very little reaction from, I think this is also a brain. This is when you insert a silicon probe in it, and you can see it damaged. It damaged the material. And so this means that these materials can couple with living matter. So rather than having a polymer here, we could actually have a neuron-like cell that would then send neurotransmitters, and these would actually influence the polymer, and the polymer would learn, now not with the ions that we send, but with the neurotransmitters of a real neuron. And this is another experiment we did with a collaborator with Francesca Santoro in Italy, where these are not really neurons, they're like neuron-like cells, but they release a neurotransmitter. When the neurotransmitter touches our polymer, if you're at the right electrical potential, it will release an electron, and this will actually change the state of this other electrode that's right here. And so the neurotransmitter has affected this electronic device here, which is just a resistor, but that's still an electronic device. So first of all, one nice thing about the polymers is, like I said, because their mechanical properties are favorable, the cells are pretty happy and they stick to them. You can see they're very, very close to the surface, so then they release the neurotransmitters. The neurotransmitters arrive at the polymer very nicely. And then they can do what is called short-term modulation, but also long-term potentiation. You can see that every time we put a pulse, there is a permanent change in the conductance of this particular part of the device, so the device has remembered that these neurotransmitters were released here. So this is a very rudimentary way of connecting living matter to an electronic device. And the last thing I want to show is that you can go even more intimate. You can actually put cell walls on top of these polymers, and the cell walls will have all the machinery that comes with cell walls, for example, ion channels. And if an ion channel is open or closed, it will change the resistance between the outside world and the polymer, because if the ions can go through, then the resistance is low. If the ions can't, the resistance is high. So you can use these polymers to essentially sense whether the cell is letting ions go through or not, which can be useful for sensing, but for a lot of other applications as well. So in this case, we show that if you have an ion blocker, then the resistance goes up compared to the initial point. If you have an ion opener, the resistance goes down, so you can really sense the ion channels opening and closing on command using the right molecules that will block or open these channels. And then the control is actually a device made with a membrane that is not able to recognize these ion blockers or openers, and there you see no change. So you can really intimately couple to biology and the very fundamental mechanisms. And so we're really excited about these soft mixed conductors. I think they can enable a new era in bioelectronics. They can emulate biology, they can couple to living matter, and they can open normal sensing modalities. And what we like is that these are materials by design, so once you start understanding the general principles of how they work, you can really design them for the purposes that you want. With that, thank you. Kurt.
So Alberto, this is really cool. How do the diffusion times compare for the natural system and in your synthetic system? And also the size of that synaptic gap.
Mm-hmm.
I mean, the distance that charge has to diffuse, how does that compare?
So the very interesting question about the timescales. The timescales are actually quite comparable. You can build circuits that will spike, so generate spikes like neuron spikes, but essentially the same timescale as neurons, because neurons are pretty slow, or polymers are pretty slow, which is surprisingly challenging for conventional electronics. So this is actually an advantage of polymers. In terms of distances, I think I can't remember, the synaptic cleft is in the order of 50 to 100 nanometers, and so that's very feasible with polymers as well. So the length scales and time scales actually match better than conventional semiconductors in many ways.
Thank you.
Maybe it's too early to talk about building a brain-computer interface. But so is anybody trying to build one with your artificial synapses? And if you want to do it, what is missing? How would it work?
So no one's really trying to build one with these materials. They're not reproducible enough yet. They're not good enough electronic materials. People are using them maybe for electrodes to sense the neural signals, and then conventional electronics will treat the signal. But I think, one interesting thought is that once you start doing this in a very parallel way, you start having a lot of signals. And if you treat them all near the brain, you'll probably melt it, just because the power needed to treat all that data. So there's an opportunity to use this brain-like computing near the sensing to be able to do low power, for example, discrimination. Sometimes you don't want to have the whole shape of the spike, you just want to count them. So maybe you can do that discrimination near the brain, and the type of circuits you can do with these polymers could be perfect to do it at very low power. So no one's doing it yet, but I think there's opportunities to combine both the sensing and simple computing that these polymers can do for low power, you would call it edge, but computing right where you do the sensing. So that if you look at people that have probes in their brains, what you don't see is that massive cable that comes out of their head and then goes to a rack of computers that does all the computations so that you can control a robotic arm. So that part maybe can be done with low power closer to the brain using these materials. But we're many, many years away from that.
Any other questions?
There's one here.
So you mean like the technology now is still, detection is more qualitative than quantitative. You can kind of detect something happens, but you cannot exactly detect like how acutely or intensively it happens for the detection.
No, the detection is very quantitative nowadays. The number of neurons that can be sensed is not so high, although Neuralink is working, the Musk company is working hard at having a lot of sensors. The problem is not so much the qualitative versus quantitative. The main technological problem actually is the duration of these probes. They go in the brain, and then they start moving, and then it's game over. And then the other problem is the computing power that's needed to then discriminate what all these different signals mean.
And like how far, like how long can they last?
Very good question. We don't know.
Okay.
Some of these are, one of these polymers is FDA approved, but the others aren't, and that's actually a very fundamental important question.
And the one you showed, the brain between the silicon ones and the polymer ones, how do you put that into the brain? Do you need to cut the slit or something?
Oops. This here.
Mm-hmm. Yeah.
So this is called a shuttle, and maybe you don't want to think about it, but it's essentially a spear that you implant into, you just jam in the brain.
Okay.
So if it's silicon, that's the spear is what you see here. With our polymers, what you can do is you can have a sphere made of a harder material, let's say a harder polymer, and on top of that you have a very thin foil, like this. And you jam the sphere in and you can peel the foil off, and the foil is the only thing that stays in the brain, which is why you don't see anything here. Right? Your probe is actually as thin as this foil. If it's silicon, your probe is going to be the sphere. There's one there.
I really liked your remark where you candid about you still don't understand how the structure is related to the properties, and you're just kind of throwing things and see if they stick. Have you noticed like any trends? Like when I've been to events at the design school, they talk about weird little things like Carl Jung's work actually kind of works out in product and industrial design in ways you wouldn't have thought of. Have you seen any trends or anything yet?
So, not really. I can tell you that a lot of papers in our community will say, here's a design rule for this, and then there's the next paper saying, here's a new design rule for that, right? So it's telling you, you don't really have a design rule. It sounds strange, but understanding how the molecular structure turns into the structure of the polymer film is really complicated, and then you throw in water with ions. What that does is even more complicated, and these materials have been around for less than 10 years. So no real trends.
So when the polymer is connected to the neuron, is there an actual bonding happening, or is it just like?
No, well, probably, I mean, the neuron will send in, I can't remember what they're called, there's some projections of proteins to kind of stick a little bit, but mostly it's just sitting on it, and there's that cleft that you saw. There is a little opening here. Right, there's a little bit of a distance here that's always there. In fact, the whole art of people that try to couple cells to materials is how do you reduce this as much as possible. So you use all sorts of topologies that the cells like to grab so that that little distance is as small as possible.
Okay, final question. Of all your research, which aspect is the most exciting for you right now?
The most exciting for me right now is nothing that I've shown here, but it has to do with the structure of it. We've developed ways to do electron microscopy on these materials. It's not freezing is not the right term, vitrifying everything in it. So you have this polymer in a liquid, and it changes its structure. But if you cool it quickly enough, everything essentially sticks in the structure that it is, and you can do electron microscopy to figure out how the molecules are arranged, to try to figure out those design rules. And it's taken us 4 years to get to the point where we can do it. So that's, to me, that's the most exciting part. It's really, it's very technical, and so not really suitable for a broad audience. But to me, it's really exciting.
Okay, great. Thank you.